Hello, good day everyone. Today I will discuss about Walker Bancrofti. So Walker Bancrofti is known for its common name, which is Bancroft's filaria. It was named after the two scientists, namely Otto Walker and Joseph Bancroft. It has two trivial names, which are filarial worm and lymph filaria. So Walker Bancrofti is a filarial or arthropod-borne nematode or roundworm that is the major cause of the lymphatic filariasis. So this Walker Bancrofti affects over 120 million people, primarily in Africa, in the Mediterranean and east-west coastal areas. In Africa, also in South America and other tropical and subtropical countries. In Asia, in coast of Arabia, India, Malaya, and north to China. So this Walker Bancrofti can result in elephantiasis, which is a condition characterized by gross enlargement of an area of the body, especially the limbs. Other areas commonly affected include the external genitals. It is caused by obstruction of the lymphatic system, which results in the accumulation of a fluid called lymph in the affected areas. Now let's move on to its general characteristics. So for the parameter description, so size range, the average microfilariae measures 240 to 300 micrometer long. Its sheath is present. Arrangement of nuclei in tail, tip of tail free of nuclei. Now for the morphology. Microfilariae of Walker Bancrofti, adult worms are long and slender with a smooth cuticle of bluntly rounded ends. Their head is slightly swollen and bears two circles of well-defined papillae. So the difference is that males are about 4 cm long and 100 micrometer wide, while the females are 6 to 10 cm long and 300 micrometer wide. So basically, females are quite larger compared to males. The cephalic or anterior end is blunt and round, while the posterior tail end culminates in a point that is free of nuclei. There is a thin and delicate sheath that surrounds the organism and numerous nuclei that are contained in the body. So for the life cycle, so as you can see, the mosquito takes a blood meal. So during a blood meal, an infected mosquito introduces third stage filarial larvae onto the skin of the human host where they penetrate into the bite wound. Next is, these mosquitoes develop in adults that commonly reside in the lymphatics. And after that, those adults produced microfilariae measuring 244 to 296 micrometer by 7.5 to 10 micrometer, which are sheathed and have nocturnal periodicity, except the South Pacific microfilariae, which have the absence of marked periodicity. The microfilariae migrate into lymph and blood channels, moving actively through lymph and blood. So the mosquito takes a blood meal. So after that, the mosquito ingests the microfilariae during a blood meal. So mag-ingest a mosquito during blood meal na sad. So pag after na na-ingest na niya, the microfilariae lose their sheaths and some of them work their way through the wall of the proventriculus and cardiac portion of the mosquito's midgut and reach the thoracic muscle. So ang microfilariae develop into first stage larvae and subsequently into third stage infective larvae. So sa third stage mo na siya ang pinaka lala na kaayo. So the third stage infective larvae migrate through the hemocoel to the mosquito's proboscis and can infect another human when the mosquito takes a blood meal. So ihara siya maka-infect if during blood meal. So maka-infect ka siya ana blood meal. So after ana, if na siya kuan, if the mosquito takes a blood meal, so there's a big possibility nga maka-infect siya another human. Now let's move on to the laboratory diagnosis. So fresh GEMSA stained blood for Walker Bancrofti microfilary serves as the laboratory diagnostic method of choice. A more sensitive method for microfilary recovery involves filtering heparinized blood through a special filter known as nucleopore filter, and then staining and examining the filter contents. Also, NOT technique may also be used. Light infections may be diagnosed by immersing 1 ml of blood in 10 ml of a 2% solution of formalin which lyses the red cells. So, after that, microscopic examination will then be performed. In all these methods, optimal sample is collected at night. Why? Because this organism generally exhibits nocturnal periodicity. So, peak hours between 9 p.m. and 9 a.m. and 9 a.m., which correlates with the appearance of its vector, which is the mosquito. So, that's the vector. However, subperiodic organisms are sometimes directed throughout the day. They are more prevalent in the late afternoon. So, there are tests that have been developed. These are the serologic tests, including antigen and antibody detection, and PCR assays, or polymerase chain reaction. So, the sensitivity and specificity of this test vary widely. So, with all these techniques available, it is interesting to note that in endemic areas gani, clinical symptoms and patient's history serves as the primary means of diagnosis. So, before you mag-diagnose, it is very important to ask the patients first, if ang sa iyang asa siya gikan, what is his or her history, asa siya gikan, patient's history, if asa siya gikan, tapos tan-awon din yung symptoms. So, that is very important to ask the patients first. So, in chronic cases with lymphedema, lab tests are usually negative. So, epidemiology, let's move on. It can be found in the subtropical and tropical areas of the world. These include Africa, the Nile Delta, India, Pakistan, Thailand, Arabian Sea Coast, Philippines, Japan, Korea, and China in the Eastern Hemisphere. Mosquito breeding occurs in contaminated water in these areas. And also, currently, Haiti, the Dominican Republic, Guyana, and Brazil are the only 4 endemic countries in the Americas. In the USA, Charleston, South Carolina was the last known place with lymphatic filariasis. The infection was eradicated in the USA in the early 20th century. So, for the prevention and control, so these are the things that we have to do in order to prevent or to avoid this kind of disease. So first is use personal protection when entering known endemic areas. So if we are already aware nga kana siya, prone kana siya or exist diha kana siya nga sakit, so dapat aware na ta to use a personal protection. Next is destroying breeding areas of the mosquitoes. Use insecticides when appropriate. Educating the inhabitants of endemic areas. Avoiding mosquito-infested areas. And mosquito netting and insect repellents are more practical and useful in endemic areas. For pathogenic effects of Walker Bancrofti, following infection with third-stage larvae, there's usually a period of vigorous immune response to the invading larvae. So, if mabot just third stage larvae gani, na instances nga, or ang atong body is mag-react siya immune response, kay nag-invade naman, so mag-response na atong body sa invading larvae. So, if that happens gani, ano na itong body, ga-react na siya immune response. Pero still, ang larvae is wala pa siya na-clear gyapon sa body na agi hapon. So, if napag-hapon siya, so the various pathologies associated with filarial infection can develop. So, diraan din ma-develop ang filarial infection. Kay, ga-react na ganun na immune response, pero still, wala pa gyapon wala ang larvae. Nagyapon siya, wala gyapon siya na-cleared from our body. So, kung na ma-develop mo din ang filarial infection. So, conditions do not appear to arise from the effects of the nematodes themselves, but from immune reactions to their presence. Most pronounced of this is the damage to the lymphatic vessels, which is mediated by the immune system's response to the adult worms living in them. Repeated episodes of lymphangitis led to the formation of fibrous and calcified tissues in and around the lymphatic vessels. So, symptoms of Bancrofti. Mostly, it affects the legs. It can also occur in the arms, breast, and genitalia. The onset of symptoms is slow, but the effects are very apparent after several years. During initial inflammatory stage, a host can exhibit swelling, granulation, lesions, and impaired circulation. Following, the lymph nodes are enlarged and dilated. They become hardened and clogged with fibrous tissue, and this prevents the lymphatic system from operating correctly. The microfilariae also cause swelling, thickening, and discoloration of the skin. Without the proper drainage of fluids, the affected tissue will expand and elephantiasis will result, followed sometimes by death. So, if dili siya maagapan, or if dili mo siya pa-check up, sagaanan nimo, so sometimes it can lead to that. So, better, if natay symptoms nga na feel, so, better nga magpa-check up. Kay dili naman siya, like, for example, mag-swell siya, daghan siya ka nang maapektuhan. So, ay mukhang kuan, wala na yung proper drainage of fluids, tapos, ang na-affected tissue, kay mo expand siya. So, daghan siya kuan, daghan siya ka nang lahi pa nga, magkay maapektuhan sa mong. So, what can be done? The parasite severe symptoms can be avoided by the use of therapeutic drugs, and protection is similar to that of other mosquito-spread illnesses. So, barriers or chemical sprays. So, dapat, in order to prevent ana, we must be very cautious. We must very careful. We must use insect repellent, singana. So, di ta kuan lang basta samot na if for example we're already aware nga kana endemic areas tapos prone kay na siya nga sakit, so dapat aware na ta sa tong self, and in order to prevent and to avoid Bancrofti. So that's all, and thank you for listening. God bless everyone.